So yesterday Alex called me and said that they're going to put ads on the, so the website that I don't, did you put the website up for eBay?
Yeah.
The eBay website, which is, it's something like clampthis.wordpress.org.
That's right.
But to fund, because there's so many people here, and to pay for all the pizza, he's going to put ads up. And so he wanted a talk title that would really get people to click on the website. So I went with one of these. So I'm Jesse. I'm a advanced graduate student in the Schnitzer Lab. And I'm going to talk about really dynamic imaging of things, so things that are transient in time and how to measure those. How many people here do electrophysiology? Is it a decent number? Okay. Yeah, but yeah, because there seem to be a broadly distributed group. So some of this is tailored more towards neuroscience type people, but there's also things tailored to, you know, the more interesting people that are imaging like protists and corn.
What are you trying to say?
You know, the brain's kind of played out, I guess. But yeah, and so I'm going to start this off with a very dry discussion of signal and noise. But the reason I'm doing it isn't just because I'm actually in the physics program and, you know, we really like to beat things into people and beat them to death, but because for anyone doing like a really dynamic measurement, for looking at things that are fast, especially optically, it's really important to think about, you know, signal and noise. Especially if you're kind of starting a new experiment and you're wondering kind of what equipment to buy, and especially if you're also kind of venturing into a new territory, which, I mean, optics is kind of opening up a lot of new applications, and sometimes it's a little unclear if the experiment's going to work. So I think it's really essential to understand signal and noise for this. And so you know, just to kind of summarize that, you know, specifically for neuroscience, optical measurements are still limited. You know, we're not picking up every spike of every cell in a region. We can still do better, and we can still develop new tools, and there's obviously a big movement towards developing new tools. So for optics, we're fundamentally, kind of at the core, limited by something called shot noise. And so just like electrophysiologists might be familiar with Johnson noise, which is kind of the intrinsic noise of a resistor, and so sometimes in your head stage you're switching between different feedback resistors if you really wanted to look at kind of single channel properties. In optics there's something called shot noise, and this is just kind of caused by randomness. So LeBron James has been hitting 60% of his shots from the field this season, right? But if he took 15 shots a night, then on some nights he's going to make 9, on some nights he's going to make 8, and some nights he's going to make 13, etc. So whenever you have like an average success rate, there's going to be variation around that. And this is given by the Poisson distribution. So here you can just see if you expected one event, then you're going to get one event 35% of the time. But if you expected 4 events, the distribution is going to become like kind of fractionally tighter around that. And so this is just a fact of nature, that you're always going to have noise in your optical measurement no matter what you do. But then to add to that, there are other noise sources. So your sample sometimes moves around, or your kind of your focus drifts, or you have a brain moving or an animal moving with a brain under the objective. Your light source is kind of going crazy, or you have read noise from your detector. But so kind of in principle you can get around these things, but shot noise you can just never get around. So there's just some things we still won't be able to do because we're not getting enough light. So the really difficult equation-y part of this is just, when we talk about signal, we talk about delta F over F. We talk about fractional increases in signal. And so the baseline noise, the shot noise on top of that signal, is given by the square root of the amount of light. But then that means that as you increase your baseline, the noise can go as 1 over the square root, right? So if you have 1 million photons, the fractional noise is going to be smaller than if you had 10 photons. And so you can kind of wrap this up into one nice equation. So, you know, say you have an action potential, or you have like a bunch of calcium coming into the cell, or I don't know what else people measure, but you know, you have sodium coming into the cell, you have a vesicle release, and you want to measure it optically. The signal noise of that event is going to be given by the delta F over F, so the signal change, the square root of the amount of light you're collecting, and the square root of the transient length. And this is kind of pictured over here. So this is from the GCAMP6 paper. If you don't know what GCAMP is, it's a calcium sensor that's genetically encoded. But what they do is they shoot off a spike in the cell, and they get a concomitant calcium transient with the voltage change. And so you can see they have a delta F over F of something like 25%, and then they have a few seconds long transient, and then they have some amount of light. And those are the things that are going to factor into, can I detect this thing? And I guess the reason I'm being kind of pedantic about this is because this is linear. So your ability to detect things goes linear with the dynamic range, but it only goes as the square root of the transient length and the fluorescence intensity. And this actually really factors into decisions you're going to have to make when you design your experiment. And right off the bat, one of these is bleaching. So as we talked about a little before, we all have had the experience of our sample getting slowly, irreversibly dimmer. And this is caused by when you excite a molecule, usually goes into some state where it then emits a photon, right? That's the good state. But sometimes it enters a metastable state, and then it starts reacting with other ion species, and then it irreversibly oxidizes or something, and you can no longer use this as a fluorophore. This is from the microscopy U site, which, as Alex mentioned, is a really good resource, like for anything you need in microscopy. I think it might not be as that, it's not flashy. It's not a flashy website, but it's solid. But the way to think about bleaching is that you have kind of a fixed number of photons you can get out of a cell. So you say 1 million, and your laser power determines how fast they come out, and that kind of goes linearly for a single photon microscopy. If you double your laser power, you're getting twice as many out, and then your photobleaching is roughly linear with the number of excitations. This isn't always true in some cases, but you could think about, if you double your laser power, your sample's going to bleach twice as fast. I think we're all kind of familiar with that, and you can kind of describe that with an equation where you have exponential bleaching, and that the rate of bleaching is inversely proportional to the intensity you use. But if you go back here, the SNR is not linear with the amount of light you're getting out, so that you have to quadruple your laser power to double the signal to noise of your experiment. And so, there's a real trade-off here where, say you can't see it, you can't see your action potentials, you can't see your calcium events, you keep trying to turn up the laser power, but you're entering a very slippery slope because the sample's gonna bleach faster than you're collecting more signal to noise. And just to give an idea that photobleaching is not the same, I guess, between all fluorescent proteins or anything. It actually varies kind of dramatically between here the red and the green, which are usually pretty good, and the yellow and the blue, which are usually pretty bad.
So Jesse,
yeah.
That then sort of introduces an idea about general experimental design, right? Which
yeah.
is to say you might want to just acquire as much signal as you can for as much time as you'd want in the ideal situation.
Yeah.
But given the fact that you have to quadruple the amount of light to get a better signal, and then compare that with bleaching, you would end up having to, say, limit the amount of time that you're actually doing the imaging experiment, particularly if you're imaging over a long period of time.
Yeah, so you're going, if you want to double the signal noise, you're going from a 60-minute long experiment to a 15-minute long experiment. Yeah, and I should say this kind of calculus is actually just kind of universal, like this kind of square root dependence. If you're also averaging over multiple trials, like this is, again, like if you do 4 times as many trials, you're going to get double the signal to noise, but not if you do double the number of trials, and so that also quickly tapers off, like the amount of boost you can get by averaging over trials. And just another kind of quick note, this is a very poorly constructed slide, but hopefully you'll bear with me. But as you sample faster, the number of photons per frame drops, right? So if you go from imaging at 10 hertz, and then you say, okay, I'm going to use Andrew Olson's line scanning confocal, we're going to boost this frame thing up to like millisecond scale frames or line scans, then you're going to kind of see a very similar calculus as before, as far as you're increasing the amount of noise as you increase, you're increasing the frame rate by a factor of 25, right? The noise is going to go up by a factor of 5. But to get the same SNR you did before, you're going to have to increase the laser power by a factor of 25. So suddenly, as you go from raster scanning your full field to line scanning, you're going to bleach the sample 25 times as fast. And so this might seem a little hazy right now, but really just, you'll kind of glean this from experience as well as you destroy samples like I have. And the solution actually, as Andrew talked about before, is image only as fast as you need to. So just like you should set your gain in the middle, you should just image at what's called the Nyquist rate, which is just kind of double the kind of signal, the maximum signal speed. So if you have a signal that's going to be like 200 milliseconds long in size roughly, or that's like the maximal time resolution you need, then sample it at 10 hertz, so 100 millisecond frames. And just the last kind of point that's kind of weirdly placed far below everything else for the aficionados is the spectral properties of shot noise. So this will be something familiar to physiologists who like to take like Fourier transforms and say that their LFP goes as 1 over f, and this is a universal, property of nature. But that spectrally, if you just had a blank sample, nothing is happening in your sample, you took a Fourier transform of the light you got back out, the shot noise is going to be white. So it's going to have a constant power. The power of the noise is going to be constant as a function of frequency. Whereas other aspects of noise, like we have line noise in electrophysiology, so you'll have a 60 hertz line, and you'll also have other sorts of noise, but shot noise is kind of white, which can be important because you can kind of filter it out if you need to. So I'm going to talk about 3 implications these kind of very dry principles have for like building your experimental setup, and then I'm going to talk about sensors and like interesting things in imaging in the brain. But so one way in which this calculus enters your experimental design is in the numerical aperture of your objective and the quantum efficiency of your detector. So we talked about numerical aperture in terms of resolution, that having a higher NA really helps you resolve smaller things. A really important thing it does is it increases the amount of fluorescence you get, and it actually goes as the square of the NA. So Andrew had this kind of light cone, and actually for like epifluorescent microscopy, it's a little different for transmitted light, but for epifluorescent microscopy, the fluorescence you get is going as NA squared. So if you go from 0.75 to 1.0, you're getting twice as much light. You start to get really big increases in signal to noise. And another note is that not all objectives are made equally. Like there's more than meets the eye in the objective. But so here's just kind of a cross-section of one that I got from either the Olympus or Microscopy U website, and you can see there's, maybe like 7 elements or something in there. And just like kind of practically, there's a few points here. One is that like there is a lot of glass inside, just like a DSLR lens on your camera. Like there's a lot of optics, and so quality is actually really important. It's not all like 0.75 NA objectives are the same, even if they have the same working distance, even if they have the same, I don't even remember what else you could have on it. There's a bunch of things, but even beyond that, there's a lot of optical properties that especially appear kind of off axis. Like you look at the center looks great, but off the axis, everything starts to look crappy. And so just like kind of DSLR lenses, like another point is that old objectives aren't necessarily bad. That there is still a lot of innovation in this sector. Like there's the NAs of like these giant objectives are kind of, this is actually I think to scale, but the NA of these objectives are kind of getting better and better, and as people have new applications like for scale and clear brain imaging, like they'll develop new objectives, but there's still oldies and goodies, and that also you can lose a lot of light in objective, so don't be surprised if your objective kind of sucks.
Jesse, do you have any crude pointers on either who makes the best objectives or parameters of objectives that tend to make them better versus worse?
I don't know. I mean, I think it's the usual suspects. Like, I think they're probably priced efficiently, more or less, as long as you go to kind of the second generation down. But okay. I don't know. I mean, we've had good experience with these giant, like $6,000 1.0 20x water ones that let you get a pipette in there. But I feel like other things like usually take the place. Like if you need more kind of clear aperture to get in a pipette, or if you want higher NA or like the immersion, but it's just something to have in the back of your mind that these are kind of complicated things. And Yeah, and so the other one is the quantum yield, or the quantum efficiency of your detector. So Andrew talked a little bit about PMTs, and he was going to talk about cameras a little bit, but we didn't quite get there, but it'll be probably online. So cameras are typically made of silicon, and they have pretty good QE. And so this actually can vary quite a bit by the camera. So you can see here's 3 CCDs, and this is the quantum efficiency in percent as a function of the wavelength.
Jesse,
yes.
What is quantum efficiency?
Sorry. So, when you have a photon that hits your silicon camera, not all photons are kind of absorbed and put into, bits. That there's actually like a loss process and like a rate of successful conversion of this photon at your camera. And so ideally this would be 100%. But as it happens, not all photons go into your camera and are successful in generating kind of information, right? And physically what they're doing is they're creating electrons or electrons in holes. But yeah, so this is this kind of just random loss. And again, we also talked about in an objective, sometimes you also have just absorption events that are again random loss. But yeah, so here, quantum efficiency is really important because you can get more light by a factor of 2 if you have a better chosen camera, right? So yeah, so that's quantum efficiency, and then it can vary, even among CCDs from 40 to 90%, which is a big margin, and so yeah, that's something to keep in mind. Like just when you're designing your experiment, you want to collect as much light as possible. That's going to give you the best chance of detecting things. And so factor this in. And I mean, even among cameras, like that's still kind of dynamically changing. Like some of you might have experience with the new SCMOS cameras that have come out. These aren't CCDs, but I think they're also silicon. But just between one generation of the SCMOS and the second generation, I think there was like a 30% change in the quantum efficiency. And so this is a big number, and it's going to really change the quality of your images. With PMTs, Andrew had a couple like side-on PMTs in his laser scanning applications. As he said, they're not that great. That they're typically kind of 20%. So this is a complicated plot. This is again wavelength and quantum efficiency on a log scale. But so here you have gallium arsenide PMTs. This is like the front of the PMT. I'll show a diagram a little later. But it's around 25%. There's better, newer PMTs, gallium arsenide phosphide, that people typically use for like 2-photon calcium imaging. That I think are fairly recent, but are now more common if you want to deal with Hamamatsu, who's terrible to deal with. But
sorry, can I just
yeah.
verify that I understand what's happening? So when the light comes out of my tissue, it's already being I'm losing some of it because it's going through some of the tissue.
Yeah.
Into the objective, I lose 79% of that comes through.
Yeah.
Okay, and then once it hits the photomultiplier, only 25% of it can get through the photomultiplier, and then I still have to do some of it with quantum efficiency when it hits the camera. So I'm ending up with like.
So you're going to use one or the other.
Yeah, use either the PMT or the camera.
That's one or the other.
Yeah.
But yeah, typically like you excite a sample, and the actual amount of light you collect is pretty low. Like the overall collection efficiency of an emitted photon is probably like 10%, under 10%. Well, that's also when you include the fact that it's going in all directions and you're only collecting a small chunk of them.
Okay.
But yeah, so there's like a real hemorrhaging process that goes on.
Okay.
Yeah, so this is another thing that came up. Where should you set your gain? And so this is specifically with like cameras and PMTs. The answer is usually in the middle. And so you're kind of when you have a chip and you're changing the gain, there's kind of actually, I say the goal here is to overcome read noise in the detector. Just because a detector has a lot of kind of stray electrons floating around, like it'll have Johnson noise if you're familiar with that. And so you kind of want to amplify your optical signal above that noise. Another thing that you could use gain for, but you probably shouldn't, is adjusting the like position of the signal within your digitizing range, right? So like your optical signal is analog. It's going to be like a current or a number of photons, and then you're going to have 256 bits per pixel or something, right? We're all kind of familiar with this. Or you're going to have 16 bits per pixel. And so you could increase the gain on your camera to kind of match the number of photons you get towards the kind of depth within this well. But the real goal with the gain is to boost it above read noise, if that makes sense. It's kind of confusing. I didn't construct this argument carefully enough. But the idea is that EMCCDs and PMTs, they use a multiplicative amplification to boost their signal above the detector level. So Andrew talked about this as well, and just to kind of go in gory detail, you have a photon that's squiggling around, and it hits this photocathode material. This is where you have this like 20 to 40% quantum efficiency, and then it becomes an electron. And then it bounces around, and at each stage you get more and more electrons that come out. And in the end you have this like very big signal. And so your goal in this whole multiplication process is to boost this above whatever kind of noise is in the detector. And this is a very technical drawing of that. So you have this gain voltage here, it's the supply voltage. And then you also have what's called the dark current, which is this stray noise in the detector, or you have your signal output. And you see that the dark current, this black line, kind of goes linearly with gain as your signal in this kind of sweet spot region, right, between 500 and 1,000, maybe around 750, 600, 800. But that you have a lot more noise at the low end and the high end relative to the signal. So we don't need to get too in the nitty gritty with this, but I think maybe the key points are that set your gain in the middle, and also be aware that there's probably other ways of changing like where you are in the analog to digital scale versus just the boosting signal above noise scale. So these are kind of separable things. And just the last thing before I move on to, yes?
Are PMTs then linear with your fluorescence? Since you're multiplying, are you multiplying linearly?
Yeah, you're typically multiplying linear. So as Andrew said, like you double, every 50 volts you double your signal?
Roughly.
Yeah, and so you can see this line here is sort of the voltage versus signal, which is actually in log space, so it's actually quadratic with voltage. So yeah, I guess that's the answer is that it's not linear, it's quadratic kind of in the voltage.
But the response property
Yeah.
is the response per photon. As I change the number of photons in, the current out.
Yeah, you can see it's not over the full range of the detector, but there is a sweet spot where you are seeing that. So just a rough rule of thumb, 50 volt change gets you a 2 volt change, 2 full change.
When you say in the middle of 0 and what's the upper limit?
So I would set it around 700 for a PMT. I mean, but within this range of 600 to 900. For an EMCCD, which is another type of camera which also has a multiplicative type of gain, it actually is again kind of above 10. You're fine. It's just then a matter of like where you are in your digitization space. Yeah.
So to be more explicit about that, the other large thing to take into account is that you have only a certain number of either a maximum voltage that you're digitizing or a certain number of a size of a well in a camera. And once you fill up that well, you saturated, or once you're above the upper limit voltage of your digitizer, you've saturated, and now you're not going to have any more response. So saturating is, of course, the worst thing you can do because you're throwing out the signal entirely.
You should just throw your experiment or something.
And I should say, certainly a lot of the prefab stuff like the Xiscopes and Liboscopes that I've worked with, they have a range indicator that the color actually represents where in that sort of either well space or PMT level you are. And so you can try and maximize your signal to be like in the middle of the dynamic range if that's where you want it to be. And you can find out when you're saturating or when your signal is too low. And that can be a really, really useful thing for getting a nice clean picture.
Yeah. Yeah, I mean, so on that, like cameras are pretty, there's like a lot of things that you can trade off within a camera. Then we're kind of touching on them a little bit as far as like well size, but I think for now, I think it's a little, there's just so much there that I'm going to have to shelf that, I think, for a little bit. But a lot of manufacturers' websites, like in their application notes too, like are actually pretty good about giving you info on like the things to think about when buying a camera. Just because, there's plenty there to occupy a very dry hour of discussion about pixel size and well depth. And,
The question, so Is the noise supposed to vary with the size of the EM gain?
So not really. You will get more, but typically if you're in EM above 10, you're typically all signal. Yeah.
Right, so if you would go from 10 to 20, let's say, and as long as you're non-saturating your signal,
you're okay.
You should have the same noise profile in both cases.
Yeah, and the noise profile should be pretty low. Yeah, or like the relative noise in that case. Yeah. Yeah, and just a note on other sources of noise. So I've talked exclusively about this very fundamental part of noise called shot noise, or the read noise in a camera or detector system. And there's other sorts of noise. I'm just going to run through them before I get to the biology. But the one thing to note is that these are uncorrelated. So different pixels, different cells for your thing are going to have kind of different noise levels. So if you average, it's going to be okay. Other sources of noise are not okay to average across. So you have correlated noise from a detector like an arc lamp. So here is a pictorial representation of a fluctuation in an arc lamp, but as arc lamps age, they degrade, and sometimes like the beam gets shifted around or it breaks up and you get like flares and nasty things in your image that are going to be correlated across pixels. And then there's other sources of noise, like you can get offset drift in the AD of your camera or in other components, like also your, warming up of like a laser or an illuminator. So just for kind of debugging this, if you think you're running into something, having like a thing of pollen grains or like a fluorescent slide around to just like calibrate is going to be like really indispensable. I mean, this would be akin to like a, what do you call that thing you plug into your head stage? The like
Model cell.
The model. So yeah, this is kind of like the model cell for an optical experiment. It's good, really good to have these around, like to debug all the sources of noise you have.
The cheapest possible way to make one is to take a highlighter and a chem wipe and highlight the chem wipe and put it under a slide.
Yeah, although
Nice.
some of the problems with those is you have inhomogeneous, like if
Yeah.
you have an inhomogeneous field, sometimes like then you're adding that in. But like Chroma will usually give you, if you stop by the Chroma booth, will give you a set of fluorescent plastic slides. But a highlighter technique is also standard. Or actually,
Slides are a really saturated solution of fluorescein, thin layer of that can work.
or whey paper too, just as fluorescent. Get a little whey paper. So part two, probably more interesting to most of you is sensors. What can we report optically? So this is just a diagram of a calcium sensor, a fret-based calcium sensor. So you have calmodulin M13, and you get 4 calcium ions that bind to it. It changes conformation, and you get fret between these. So you see a signal increase. And so this is kind of the physical mechanism that goes in. One important point also with protein sensors, especially, is that the kinetics change. So this is a fluorescent voltage sensor, and so you're having a stepwise depolarization or hyperpolarization, and the kinetics of on and off are non-trivial, especially if you're trying to look at things kind of on a millisecond scale. So you kind of want to know what you can report and how fast you can do it. So there's a lot of sensors out there. I'm not listing all of them here, but I think this is just kind of a grab bag of some of them. I mean, you can look at calcium, so there's dyes and fluorescent proteins that are available. And the dyes are usually pretty good, but harder to work with, just kind of as a rule. And so you get those for calcium and voltage, and definitely ions as well. There's now glutamate probes, which I include just because I wanted to say glue sniffer. And you also have monoamines and pH, and you can see vesicles releasing. So there's kind of a wide world of things to look at. And what you probably already know from what we've talked about, this is just kind of a brief recap, is that the things that are really important are dynamic range, and also just the on-kinetics. In part because this will match the dynamic range. Like if you have a sensor that takes a really long time, it takes 500 milliseconds to turn on, but your transient is only 200 milliseconds long, then you're not going to get that full dynamic range of the sensor, if that makes sense. Then brightness is obviously like really important, but secondarily to dynamic range, and the off kinetics, which we'll talk about in a second, and the spectra, and then also the photostability. And just to add to Alex's kind of list of things, you can get good kind of lists of optical properties of things like excitation minima, or and absorption maxima and extinction coefficients and quantum yields on the like Nikon Microscopy U website. Roger Chen's website also has a lot of like the more nuanced fluorescent probes, like your bananas and your strawberries, and like the spectra of those. And this Nature Methods paper down here, this Drobazev et al., actually has, I think, the best listing of 2-photon absorption spectra, which are, for a lot of the in vitro dyes are kind of on their like wet spec sheet, but they can be a lot harder to come by because I think, in part, that I think they're a lot harder to measure. So this is actually kind of I think a very useful resource for a lot of people doing 2-photon.
I should say for 2-photon too, there's also the Dr. Bio website. So if you Google Dr. Bio, DRBIO, it's Wattweb's website at Cornell.
Okay.
He's got a bunch of cross sections for 2P as well.
Yeah, Wattweb is one of the godfathers of 2-photon microscopy for those not in the know. So off kinetics, it's a double-edged sword. It's a frenemy. It's GCamp S versus GCamp F. So obviously, if you have a longer time to integrate signal over, so this big black line, you're going to get a much bigger signal noise in your transient. It's going to be a lot easier to see. But obviously, if you had one calcium event at 0 and one at 1, it's going to be a lot harder to resolve those two different ones with this off kinetics. So with that, I want to just show a movie that's going to bring us more into the physiological imaging territory and the types of approaches people are taking. So this isn't going to be kind of as boring or comprehensive as the previous part. But so I'm going to kind of use GCamp, which is a genetically encoded calcium sensor, just as an example of the kinds of things people are doing with activity-based imaging. And so this is just a 2-photon stack sped up 10 times. And so what you're seeing is these are all a bunch of cells, these little annuli, and that's their cytoplasm. Their nuclei is kind of dark and here. And what you're seeing is a 10x speed up of cortical activity that's ongoing. And so you can see there's a lot of flashes, and you see like axons, and you have really nice spatial resolution. And so I'm going to talk just a couple notes on GCamp, since I think a lot of you from neuroscience are probably interested in, or may be interested in working with this calcium sensor. So here's a very, very low resolution crystal structure. I think this is like a 20 angstrom level crystal structure of the bound form of GCamp. But so it's also based on a calmodulin M13 kind of exponential change. And just one note is that with any fluorescent protein, just any fluorescent protein that you're expressing, obviously getting the right amount of expression is really critical. Even if you're just having a tag, sometimes if you overexpress, things will dimerize. Or if you have too much archirhodopsin or heterodopsin, you'll get aggregates of misfolded proteins. Or with GCamp, the particular failure mode is that long-term expression or overexpression can cause cytotoxicity. And this manifests itself as calcium entering the nucleus, 1. And number 2, you have decreased response. So this is kind of hard to see, but down here you have the number of action potentials and the delta F over F of, I believe, a calcium dye they put into these filled or non-filled cells. And you can then look at the change in fluorescence, so as a readout of kind of the change in the calcium that's binding that's occurring in the cell. And you can see that neurons that have nuclear fluorescence versus those that don't, you're getting a pretty big difference in dynamic range. So this is bad, and you should not overexpress things. And so choosing the serotype of the virus if you're working in animals, or the promoter of your virus is obviously the utmost importance. And another note with GCamp, and this is also for any sensor, it's still kind of not clear how well GCamp works in a lot of cell types, particularly interneurons. And one reason for this is that, with calcium, neuroscientists kind of rely on the fact that the intracellular calcium inside a cell is usually very low, as opposed to something like sodium and potassium. So the reason it's really easy to look at calcium is that there's giant, like, fractional changes in the calcium concentration. But the actual resting potential, so this is the molar concentration of calcium, so typically around 6 or typically around a micromolar or below, and the change in GCamp signal you get as a function of changing the calcium concentration inside the cell. And so this is from a paper where they generated a whole bunch of mutants of GCamp that have different calcium binding affinities, but you can see that you can move this around, but this type of affinity of GCamp or any sensor you have for the protein is like really important. So you really have to think about the details whenever you're doing kind of one of these experiments. And then obviously with GCamp specifically, it's not clear how spikes and activity then like match up with all the different cell types. And here's just an example of the calcium you get from a single spike. So part 3, I'm just going to run through. Yeah.
Actually, apropos of that, I don't know how similar it is with protein sensors, but I imagine it would be similar to what can happen with dyes. Is that how it performs in a cuvette and how it performs in the cell can be very, very different, because the dyes can bind to things in the cell, and it can change its fluorescence properties, can change its calcium binding properties. Yeah. So that kind of thing could be happening.
Yeah, no, definitely, and especially like as you move from like even cultured cells going into like tissue, you're just like, you keep taking hits, where you start off with, this beautiful 30% delta F over F for a spike, and then suddenly you have like 1% transients. So, yeah, but I guess that's more preparation, and you're talking about more in the physiological arena.
Yeah, so I mean, that could explain why things are happening in it. Different things are happening in inner neurons. Maybe there's proteins expressed in the inner neuron that are binding to the GCamp and changing its properties.
Yeah. Yeah, there's a lot that goes on once because it gets complicated. Yeah, so part 3 is not this type of in vivo imaging. I'm going to talk about cellular resolution in vivo imaging, but I think mainly the in vivo part, I just mean talking about tissue, talking about like when you're actually in the sample of interest. So this could even be like a plant, or I guess, or protist, or a zebrafish. But obviously there's 2 challenges. You have to keep the animal happy, you have to keep the optical resolution there. And there's 2 solutions that have predominantly been pursued, which is head fixed 2 photon and freely moving 1 photon. This is not exclusive, but this is the kind of paradigm I'm going to draw for pedagogical purposes. So scattering, we've kind of talked around this a little bit. Brain tissue, any tissue scatters light. It does not absorb light really. So on the bottom, I have the wavelength versus the relative absorbance of water, which is what 70% of our body is. And you see that this isn't a log scale, so 0.1, 1, 10 arbitrary units, but you see that there's this big window between kind of 500 and 1,000 nanometers. So right where you're imaging, where there's no optical absorption. But there is scattering. So this is simulations of scattering in tissue as a function of wavelength, again, in log units. And you can see that there is this big taper off as you get into the infrared of the amount of scattering you get, but it's actually still fairly significant. And so so that kind of gets at the wavelength dependence of the scattering. To actually illustrate that, this is from a review from the D Lab, where they just crammed an optical fiber into tissue and shot light in, or they did the same thing in saline. And you can see with saline, you get kind of a nice distribution of light. But in brain tissue, you kind of get this conical thing because the light's just kind of going all over the place. And so the kind of practical consequences is if you're in tissue, you have to turn up your 2 photon power as you go deeper, and that for 1 photon, the resolution is just going to get worse and worse and worse as you go deeper and deeper into tissue. So for looking in a mouse or an organism, there's kind of the simplest prep is to just cram a cranial window onto him. And so this is basically taking the skull, and there's a lot of ways to do this, but basically you open up the skull, you jam on a cover slip, and you make sure to put, you have to usually take off the dura or not take off the dura, and you then have to wash and wash and wash because it'll be bleeding, you don't want bleeding. But then you can fix this cover slip down, make sure you put pressure on the brain. And this can actually stay optically clear for weeks and weeks and weeks. And so this is kind of a standard prep. It works in the spinal cord. It works for even deeper brain structures. You can work around this. There's definitely challenges, like you get inflammation, you get bone regrowth, you get there's a lot of messy things that happens when you work in animals, but it is kind of fairly standard. And you can start to get GCamp expression that looks like this, where you see, again, a bunch of cells that all look nicely annular. And then you can, just kind of cram your mouse on a ball under a head holder and image his brain. There's also the complex approach that has been pioneered by Forrest's old lab and Forrest himself. Forrest, were you responsible for this?
I'm partly responsible for this.
Forrest is partly responsible for this. But basically, you also have a mouse on a ping pong ball on a hairdryer in front of a toroidal screen with a projector shooting at like simulated trees and really ethological stimuli.
I'm definitely responsible for the art that's on the walls there. Actually.
For the trees.
Yeah.
Yeah. So this is from Quake, the Quake video game.
That's right. Yeah.
So Forrest decided that Quake was more natural than like Mario 64, I guess, for the mice.
Yeah.
But you have this giant. What is this screen? Is that just?
The screen is a custom-made torus.
Okay. So
made of semi-reflective material.
So you have a semi-reflective torus. But basically, so there's a lot people are doing now with behavior, and obviously this is not super simple to set up and get working. I mean, you have to get the projector, and then they had some crazy closed loop system, and now people are still working on this, and it's super cool, but also has a higher barrier to entry than just the cramming the mouse on the ball, I guess. But so this is certainly like an approach that people are going to, and I think like a lot of labs have set this up. And then kind of down here you have the same situation where you kind of have a glass cover slip and inside some kind of cannula and head bars. And you kind of make it, and so that's the basic idea. Chronic versus acute. You kind of make choices, like obviously if you have an acute prep, the reason you're doing it is so you can jam a pipette or an electrode into the mouse brain. And so typically there you won't use a full cover slip, but you'll kind of have your cover slip, and then you'll put agarose on top of the brain, then you put like enough cover slip on top to put pressure on the brain so it doesn't just kind of expand. The brain's under pressure. So the brain will mushroom out if you don't put pressure on it. But the agarose is there so you can then like jam a pipette in the brain and torture yourself for 6 months. So these are the kind of 2 photon approaches, but I think that they're relevant for people with a lot of different preparations. Just that especially for like rodents, the kind of surgical techniques I think are really there for imaging superficially. I want to talk about something our lab does, which is freely moving single photon. So to contrast the, really nice resolved cells you saw before, our lab has used kind of miniaturized microscopes, and so these are single photon microscopes, and what you see here is a mouse running around, and then you also have flashes in part of its brain. And so these are cells. You can see that there's a little more crosstalk between cells. They're not as sort of nicely resolved, but you get the idea. I mean, I'll go more in depth in this in a second, but the basic idea is that calcium sensors are very good, and so that you can kind of use preps that are poorer optically to look at activity. And to get more in detail on that. So as opposed to this picture of nicely lit GCamp cells, the green here is the nicely lit picture of GCamp cells you see from a single photon microscope, I believe. I think this is just like a Z projection or something. This is with GCamp 3, so things get better. But then you can still get pretty nice calcium traces from individual ROIs within here. and you can resolve a lot of cells. And so one of the advantages that single photon microscopes have, this is a trade-off I haven't really talked about, but that you get a lot more cells at the same frame rate just because you don't have to raster scan. And so kind of proportionally, depending on the scan and things like that, you typically can get a bigger field of view at a faster frame rate. Obviously there's cons here that you have lower spatial resolution. You have optical aberrations with a miniaturized system. Like you make trade-offs when you design small things, and you get more crosstalk between cells and arguably lower signal noise between cells. But you're also collecting kind of more light. Two photon is in some sense also a double-edged sword because you're getting really nice spatial resolution, but you're only at a specific depth. Whereas single photon, you have a much deeper depth of field, which means you can collect light out of all of them if you're interested in cells at multiple depths. This is not the only freely moving approach. I'm biased in this case, but there are many fiber optic and kind of fiber bundle approaches that people have pursued at looking at activity in kind of freely behaving animals. And obviously you can use one photon in this kind of preparation as well. It's just kind of less established. Two photon, I won't dwell on this, but you can get axons, you can get dendrites, you can get spines on dendrites, as Andrew mentioned, you can get vasculature. Two photon also has just kind of a bag of tricks associated with it. There's a lot of really nice methods, some of which you can see here, that are usually fairly technical, I think, for non-optics labs, but there really are ways of pushing two photon to for example, being very fast and randomly accessing different points. And there's also FLIM settings where you could use fluorescence lifetime measurements that are, in principle, a little more informative than just changes in fluorescence. There's also deep brain approaches that I'm not going to dwell on, but basically you can suck out part of the brain and you can put in part of a lens, or you can put in a very small lens and use that to relay light from this area of the brain to up here. And so then you can use that to chronically image deep brain structures that you can't get just with the cranial window. Since the two photon, I didn't actually say this explicitly, but you're kind of in the 200